In an earlier lesson, we referred briefly to the third base wobble. Let's now see how this relates to tRNA recognition of the codon. Transfer, or tRNA, is truly the translator in this process. Each tRNA carries an anticodon, a sequence of 3 bases, that binds to the codon in the mRNA message through complementary base pairing. Since the code is redundant, does this mean that there must be 61 different tRNA molecules to recognize the 61 codons? We find that as there is a wobble in the codon, there is a wobble in the anticodon. Keep in mind, the binding of the tRNA to its codon in the mRNA is antiparallel in orientation. This means that the third base wobble in the codon base pairs with the first base in the anticodon of the tRNA. We find that many tRNA molecules can recognize more than one codon through variations in hydrogen bonding. This means that we need fewer tRNA molecules than there are codons. We can see this wobble well illustrated in the figure on the right. The third or wobble base in the codon pairs with the first or wobble base in the anticodon. The wobble base in the tRNA can base pair with more than one base in the codon. Here we see illustrated several examples of non-canonical base pairs that would constitute this wobble pairing. On the top left, we see examples of pairing between G and A and between U and C. On the right, we can see U-U base pairs. In some cases, the tRNA carries a non-standard base, such as inosine. Base pairing with inosine is shown on the lower left and lower right. Let's remind ourselves of the benefits of the degenerate code and the wobble hypothesis. It means that we need fewer unique tRNAs, and that misreads of the code will not necessarily result in mutations that damage the cell or organism.